Hello, the article that we're going to be talking about today is called The Spatiotemporal Dynamics of Microglia Across the Human Lifespan. So, we're going to be talking about microglia, which is basically the macrophages of the brain, and they derive from the from the mesoderm. So, as you can see here, this is the post-conceptual week, the embryo life starting from week 0 and ends at birth here about week 38, and then this extends from birth to the age 75. So the green color here represents the microglial proliferation. So as you can see, and the purple color is for the microglial density. So as you can see, there is, for the proliferation, there is two main waves. Here, this is the first wave, and this is the second wave. The first wave happens about 9th post-conceptual week, and the second wave starts about 18 post-conceptual week. So as you can see, for every proliferation wave, it's followed by a density wave. So that will make sense. So this is the first density wave, and this is the second wave. And in the adulthood, um, the microglial densities and proliferation is kind of stable, so they remain at the baseline level. So here, this is a cross section of the embryo at the third post-conceptual week. It's a mid-sagittal section, and we are taking three samples. We're taking samples from the brain, a sample from the endoderm, and a sample from the heart. And we're using different chemicals to visualize the cells. So here we're using the IBA1 Ki67, which is a stain for the proliferative microglia. And as you can see, we don't see the stain here. So that means the microglial cells are absent everywhere in the embryo at this stage uh, of the embryo, third post-conceptual week. And when we were using the PUI stain, which is indicator for the myeloid lineage, which of course include macrophages everywhere, we can see the presence of the macrophages in the endoderm and the heart only, but we don't see it in the brain. Here we are following up cut sections of the embryo again. It's from the fourth post-conceptional week until the 8th post-conceptional week. So we can see, and here's it stands for the mantle layer, um, and here is for the ventricular zone layer, and this is for the telencephalon, the brain, and this is for the liver. So we can see that based on this stain, that we start seeing the microglial cells starting from fourth post-conceptional week. And we can see that the microglial proliferation is restricted to the mantle layer only. We don't see it in the ventricular zone. And in the liver, we can notice that the proliferation is highest at the sixth post-conceptional week, and it's lowest at the 8th post-conceptional week. And when we look at the morphology of microglial cells, when we look at it in the mantle layer, we notice that the migratory phenotype is tangential. It's not radial, and this is tangential, so it suggests expansion within a layer. But when we visualize it in the ventricular zone, we notice that the migration phenotype is radial. It's radial like that, so that indicates the colonization of adjacent layers. And as we mentioned earlier, there are two waves um, that for the peaks of the microglial cells. So the most relevant time points macroglial disease, the first wave we see at the fourth post-conceptional week, and it indicates colonization of the brain by these macrophages. And the second wave is at the ninth post-conceptional week, which indicates a transition from embryonic to early fetal life. When we compare the proliferation and density between the brain and liver, we can see that the brain proliferative index is much higher in the liver, but the density is significantly lower. So these two doesn't have to do with each other. And here we're taking different samples from the embryo. The samples in brown is a sample from the forebrain, and the samples in orange is from the hindbrain. This is from the fourth post-conceptional week. So if we compare these two samples, we notice that there is a few macrophages in the forebrain, whereas the density is much higher in the hindbrain. And if we look at here, we notice that the macrophages are entering the brain parenchyma from the ventricular zone, the meningeal compartment, and the ganglionic eminence. Here is we're taking a coronal section from the embryo. We can see the different layers of the brain during development from 9th post-conceptional week until the 12 and a half post-conceptional week. And it's worth mentioning that the 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 pre-subplate transforms into a subplate start at the 12 and a half post-conceptional week. So again, here the two waves we were talking about. So the first big wave is at the 12th post-conceptional week, and after that we see that the cells start to decline they start to die by apoptosis. So we are using the caspase 3 stain and we saw the cells starting to die. And we see the same thing in the second big wave at about week 20 to 22. We can see the cells starting to die and decline in their numbers. And so it's worth mentioning that between 9 and 10 post-conceptional week, we detect a significant transit switch in the migratory phenotype in microglia. It changes from tangential to radial phenotype, and this suggests local colonization process being coupled to cycles of expansion. So here we're talking about as a marker called TMEM119. It's a hemostatic marker for microglia. And by the way, there are conflicting reports in the literature about its validity as a bona fide microglial marker. So when we look at the cross section from the 13th post-conceptional week, we're using the TMEM119 dye. We notice sporadic expression of this marker was detected in the lower portion of the cortical plate, but not in any other middle layer. And when we use the topographs, we can see the TMEM119 plus cells are strongly in the marginal zone. You see the green color, and we also see it in the ventricular zone here. So here we are looking at the spatiotemporal dynamics of microglial cells. Spatiotemporal means time and place. We want to see about the microglial cells. So we're following so the temporal it means the time from the 9th post-conceptional week until the 36th post-conceptional week. And spatial, which means about the place, we want to see, are they on the top of the developing cortex, or are they on the bottom cortex? So earlier in the post-conceptional week period, from the 11th and 12th post-conceptional week, we see microglial cells. You see here the heavy red color. They are at the top of the column, at the top of the developing cortex. So they are in the marginal zone, cortical plate, and pre-subplate. So here we see them on the left, but we don't see them late on the right. Whereas when we look at the late embryonic stages, specifically week 21, week 23, and week 25, you can see the microglia are present in the lower segments of the developing cortex. So we see it in the subventricular zone and the ventricular zone. So here we see it at the right. We don't see them early in the left. Here we have this diagram. They call it a volcano. On the right, on the positive end, we see upregulation of these genes, these three genes, and these are associated with chemokine and toll-like receptor signaling. And on the left, these negative values, it indicates downregulation of these genes, such as TGFBR1, and they are associated with cytokine receptor signaling and interferon gamma mediated signaling. Thank you so much.